The EU Iris-Cov proposal was created as a response to the EU Emerging Funding Action that was launched in March 2020, trying to find solutions for the COVID-19 pandemic and focusing primarily on epidemiological studies, the development of diagnostic devices at the point of care, and the development of novel vaccines and treatments. For this reason, and because of the emergency of the situation, the EU was looking for solutions that would provide a high technology readiness level answer. So Iris-Cov belongs in the molecular diagnostics area and proposes to develop a system that will be simple to use at the point of care for the development of SARS-CoV-2. The whole concept is based on a lab prototype developed already and named Iris, which operates by doing isothermal amplification on the LAM method and then combined with colorimetric detection. Someone can detect in less than 30 minutes the presence or not of the virus in patient samples. Advantages of the method are that it's a very simple method, can be used by minimally trained personnel, and only relies on the use of a smartphone or tablet, and of course takes advantage of connectivity to transfer data from the point of test to a main decision center. The Iris prototype was developed at Fourth Lab and was further improved significantly and exploited by Biopix DNA Technology, a spin-off company created for the purpose of exploiting and investigating the Iris concept. So within the 18 months of the project, our aim is to develop a final device that will be of an industrial level and will also be certified in order to be disseminated directly to end users and to those who need it. In addition to Fourth, other partners include the device manufacturer, Biopix DNA Technology, the enzyme biotechnology, EnzyQuest, a certification partner, a very important partner for this consortium because they will lead through the consortium to the final certification and product, PK&M Solutions, and finally 3 end users, 3 hospitals who will test and certify the validity and the clinical importance and performance of the system in a clinical hospital.
The main challenges of reaching the market so fast were that, first of all, none of the researchers had experience in building a product, a final product, and also the regulatory framework, the demands of the regulatory framework, which have been very demanding and are becoming even more demanding as we are phasing from the in vitro diagnostics directive into the in vitro diagnostics regulation. We need to resubmit the technical file for the product and reach a level that it is compliant with the very strenuous prescriptions of the regulation.
EnzyQuest is a biotech company was founded in 2019. We are also situated in the Science and Technology Park of Crete. And our goal is to develop, manufacture and commercially exploit reagents and enzymes that their applications in the molecular diagnostics. Our role in Iris-Cov is to manufacture the molecular tools, the reagents that are needed in order to detect SARS-CoV-2 using the Pebble device. So we actually succeeded that in collaboration with Biopix and the Institute of Molecular Biology and Biotechnology. And now we are proud to say that multiple test points can be actually established in airports, diagnostic centers, ports, etc.
So in our immediate plans is the development of further compatible test kits for the Pebble platform, both for infectious diseases and pharmacogenomics. And for this purpose, we are in the process of raising additional private capital, but we have also reserved part of the necessary funding to be submitted as proposals for the EIC funding, for which we hope we're going to be successful.
Now, a main advantage of our technology is that it's very simple to fabricate and very cost-effective, making it an ideal solution for the developing world. For this reason, we have also embarked in the consortium, an SME partner in South Africa, Cara Health, who in parallel will develop, manufacture and certify the whole system in South Africa with the aim to release it to sub-Sahara areas and therefore provide a solution for addressing and managing COVID-19 testing and spreading in both the Europe and the Africa continents. Through this project, we expect to develop a finally certified prototype that will be easy to use at the point of care straight away, meaning where the patient is or the sample is, such as airports or old people resident houses or schools or barracks. The main challenge within the project is to manage to create this certified project within just 18 months. And another milestone is achieve the development and production and release of the final product in both Europe and South Africa. Something very difficult to achieve in such a short time, but of very high impact with both social, economic and health implications. However, we are foreseeing to use this device not just for COVID-19, but for other viruses or pathogens detection. And we're also interested and believe that this technology will be very suitable for detecting foodborne and plant-borne pathogens in similar samples. That means not just human samples, but also plant and food tissue, and always at the point of care or at the field or where the sample is.